Welcome to the Cambridge Neuroscience Interdisciplinary Seminar Series. This series features current work from across the schools and departments of the University of Cambridge, reflecting the pioneering work and diverse interests of members of Cambridge Neuroscience. Cambridge Neuroscience is currently going through quite a detailed consultation process to develop 6 new themes for the research we do here. Each of the next 12 talks will come from one of the 6 new themes, 2 from each. For more info on the themes and the talks covered in this series, please follow the links below and follow us on Twitter at CamNeuro. So welcome everybody to the last in the current series of the Cambridge Neuroscience Interdisciplinary Seminar Series, where we're delighted to welcome Stefano Pluchino. Stefano is a university reader in Regenerative Neuroimmunology and honorary consultant in neurology within the Department of Clinical Neurosciences here at Cambridge University. He has a strong interest in regenerative neuroimmunology, and that is what he's going to speak to us today about in the context of stem cells. Over to you, Stefano.
Thanks so much, Elvira. I'm delighted to be with you guys this afternoon, and I will be very happy to share with you the most recent progress in my lab has made towards the understanding of how to identify the principles of regenerative immunology with a specific focus towards next generation stem cell medicines. But before we get into the real scientific data, I think it's important to provide you with the appropriate context, and it is important to clarify what is regenerative neuroimmunology. To do so, we have to start from the concept that the immune system is absolutely indispensable to protect us against pathogens of any type, including viruses and parasites, but also cells of the immune system play a major role in promoting tissue healing after different neuroinflammatory conditions, including multiple sclerosis. That stem cells are, of course, indispensable for tissue and organ development, but they're also important for reparative regeneration in a number of tissues. And in the brain specifically, stem cells are being actively investigated due to their potential for clinical translation in different conditions, including pure neurodegenerative, but also more complex neuroimmunological disorders, including especially MS. Further to this, there is also an increased recognition that the immune system regulates brain development by modulating generation of new neurons, generation of new malinforming cells, and formation of new synapses. And therefore, the understanding of the basic mechanism for the interaction between the immune system and the brain hold the real promise to enhance our ability to manipulate the immune system in a way that will be improving repair and interfere with neurodegeneration. And the importance of all these interaction and principles is, of course, highlighted in the search of next generation regenerative therapies in complex neurological diseases. At the beginning of our journey towards regenerative neuroimmunology, there was great expectation that using and manipulating exogenous transplanted stem cells, or even fostering the endogenous stem cell compartment in the brain, we would have been able to promote the creation of circuits between the graft and the host, implying neural and non-neural cells in the host, but also the creation of new circuits between the host and its whole and the endogenous stem cell compartment. And the disease we selected as disease out of which starting making our first observation was MS, which in theory is one of the biggest challenges for developing a regenerative medicine approach in a classical way, for a number of reasons, even though there is significant potential behind MS. MS, in fact, is a chronic disease of the central nervous system, which is autoimmune in nature. It is more prevalent in females as compared to males. It is clinically pretty heterogeneous, with fluctuation of relapsing, remitting, and progression. It is characterized by inflammation, demyelination, and degeneration of the brain. And there is significant interest towards the endogenous, intrinsic reparative potential of the MS brain because of the innate capability of a number of these white-appearing lesions at the MRI to undergo spontaneous repair. In addition to this, what is highly relevant nowadays is the significant availability of disease-modifying therapies, which have been developed by different companies, to approach, therapeutically speaking, the most frequent form of MS, which is a relapsing remitting form of MS. And you see here a long list of approved disease-modifying therapies and hypothesized mechanisms of action. However, despite 85% of people initially diagnosed with MS, at the time of diagnosis develop a relapsing remitting form of the disease, the great majority, if not all these disease-modifying therapies are practically unable to inhibit or to avoid the transition from an early relapsing remitting clinical course to a delayed secondary progressive disease course. And there is evidence that in the late stages of MS, when the disease becomes progressive, part of the accumulation of secondary damage in the brain, which becomes irreversible in nature, is the result of different compartments of mononuclear phagocytes, especially blood-derived CNS-infiltrating macrophages and locally recruited microglia to damage selectively malignant sheets. And this is, of course, a working model, which, however, is receiving very, very interesting and compelling confirmation in humans. And as you can see here, by applying a high-field in vivo 7 Tesla phase magnetic resonance imaging in people with early, mid-stage, and late disease, this group from the NIH has described a strong association between chronic active, smoldering type lesions in the brain and accumulation of irreversible disability during progressive MS, therefore implying the description of a new biomarker for testing a new generation disease-modifying therapies. Within this context, which is kind of background of our conversation today, the worldwide initiative, which is called International Progressive MS Alliance, has identified a few years ago, and it is still valid, a number of key areas where to invest in terms of funding, ideas, and careers, one of which is the development of new therapeutic agents that, for being successful in the context of progressive MS, hold the great ambition and the great responsibility to be able to reduce inflammation once it becomes compartmentalized into the brain, to provide combination of neuroprotection and health restoration of lost function to increase plasticity and repair. And I guess you agree that these 3 major features all together included in a single new therapeutic agent are very, very, very unlikely to happen, if not considering new generation molecular medicines. As such, developing stem cell medicines in neurology might really hold the promise of encompassing all these expectations together in a single approach. And in general, when thinking of developing stem cell medicines, or at least non-hematopoietic stem cell medicines for brain diseases, one can think of different strategies. On one side, one can think of rescuing endogenous progenitors around areas of the brain where damage is taking place, aiming at a type of regeneration which is called reparative regeneration. Or one can think of mobilizing endogenous multipotent or variably potent progenitors from prototypical germinal stem cell niches, aiming at making new neurons or making new oligodendrocytes or making new astrocytes. Of course, it is still very exciting proposing to transplant exogenous stem cells able to travel in the brain and to reach those areas where some protection and restoration of tissues is needed. Or the future is for in vivo reprogramming post-mitotic cells into each desired type of interest, cell type of interest, to promote reparative regeneration. So let's start from strategy 2, which I believe always in the context of regenerative neuroimmunology is really opening new avenues of understanding the biology of stem cells and therefore anticipating new ways of potentially manipulating the endogenous stem cell compartment. So one first evidence I wanted to share with you is that the advent of next generation ex vivo single cell RNA-seq has revealed in the adult subventricular stem cell niche the presence of a discrete population of highly glycolytic and highly enriched into lipid metabolism, dormant neural stem cells, a type of stem cells which are very flexible in nature and can rapidly undergo activation. So these dormant NSCs are also characterized by high protein synthesis and are able to differentiate after priming into activated neural stem cells. And what is interesting is that cytokines, pro-inflammatory cytokines, interferon gamma, which initially was believed to be irrelevant in the brain, or at least unable to signal to stem cells of the brain, is able to induce this activation from dormant into active or activated neural stem cells in response to acute brain injury. So this is the first evidence of the kind of promiscuity of signaling between the immune system and the brain. There is also another very exciting paper which came out a few years ago showing that stem cells of the brain, especially those, the progeny deriving from neural stem cells, which were initially thought not to have anything to do with the immune system, are instead characterized by this intrinsic capability to exhibit phagocytic activity in vivo and ex vivo. And you see here this very nice evidence in vivo after phosphatidylserine functionalized liposomes were injected intracerebroventricularly. And the inhibition of phagocytosis in the neurogenic niches, both in the dentate gyrus of the hippocampus as well as in the subventricular zone, is able to inhibit neurogenesis. And the development of ELMO1, which is a specific gene controlling phagocytosis, loss of function mice, shows the accumulation of apoptotic nuclei in the neurogenic niches, and as a consequence, a significant impairment of neurogenesis. Another initially immune-like function instead now found on stem cells of the brain. And there is also another cytokine, again, pro-inflammatory Th1-like classical cytokine, TNF-alpha, which is able to induce activation of neural stem cells and also to return to quiescence by signaling with two alternative cognate receptors, TNF receptor alpha, TNF receptor 1 and TNF receptor 2 on neural stem cells. That systemic inflammation is able to reach the brain and to signal into the brain through TNF-alpha signaling, and signaling through TNF receptor 1 is able to promote the return to quiescent of otherwise activated neural stem cells, and instead signaling via TNF receptor 2 is able to activate neural stem cells in prototypical germinal niches. And this is something not very far away from what we in the lab showed a few years ago when challenging the idea of flexible and plastic endogenous neural stem cell compartment in the context of experimental nephropathy as a model of MS. In that case, what we found, which was completely unexpected, was that after the induction of MOG-dependent active EAE, instead of observing a significant response of the endogenous stem cell compartment towards either generation of new neurons or new oligodendrocytes, instead we found a significant dysfunction of the generation of these runners, these PSA-NCAM new reactive running neuroblasts, which every day are generated by subventricular zone resident stem cells, and they migrate to the olfactory bulb in adult rodents, which accumulated at the level of the lateral ventricles. And we also showed a significant deregulation of cell cycle features in neural stem cells where Th1-like pro-inflammatory cytokines, including interferon gamma, IL-1 beta, and TNF-alpha, the very same cytokines I just described to you in other studies, were able to induce P21 cyclin-dependent kinase inhibitor 1A, induction of exit from G0, G1 phase, and from S phase, and induction of quiescence. So again, another evidence of cytokine-dependent control and regulation of physiological neural stem cell function during pathology. And then we have also this very recent compelling paper showing that there is also something to consider, that as long as we get older, or as long as our animal disease model becomes more aged, stem cells of the brain become progressively unable to keep their homeostatic function and exhibit molecular aging. This molecular aging in this paper being described as decline of the number of bona fide neural stem cells in the typical germinal niches, increased quiescence, and upregulation of ABL1, which is a gene whose role is not yet fully established, but it is argued to be interacting with other aging-related genes, controlling DNA damage, accumulation, and mitochondrial function. As a matter of fact, when inhibiting ABL1 with imatinib, which is a commercially available FDA-approved medication for cancer, we are able to restore neural stem cell function in the middle-aged brain. So all this to say what? That there is a very complex underlying interaction between the systemic circulation, the immune system, and also the physiological aging of the individual. And in fact, I was very excited to read this paper that Alex Incase, at that time a PhD student in Sid's Crockett's lab at the University of Connecticut, but now a postdoc in my team in Cambridge, showed a couple of years ago, when she described using iPSC-derived disease modeling with stem cells, significantly faster aging of stem cells in the PMS brain. And she started with this observation where she described a significant number of P16 expressing SOX2 bona fide neural stem cells, progenitor cells, in the context of the MS brain compared to control, which increased with the age of the lesion. And then she moved in vivo by accessing patient-specific and control iPSC lines and inducing progressive differentiation into stably expandable cycling neural stem cells, and subjecting patient and control neural stem cells to a number of targeted approaches to check for senescence, and also much less targeted proteomics, observing, first of all, confirmation in vitro of a significant upregulation of the cell cycle regulators, which become predominant during aging, P16 on the left and P53 only in Cambridge. She was able to independently replicate and confirm early data using a technique which is called direct induction into stably expandable brain-specific stem cells, which is much less of impact for the biological clock. So when looking at the age of a cell, it is much better to induce a partial reprogramming from somatic fibroblast into tissue-specific brain stem cells, rather than induced pluripotent stem cell technology. Also using independent NSC approach, Alex has been able to show a significant PMS lines, but only in control, which is also associated with the increased expression of gamma H2AX, which is another marker of aging. Again, a significant difference. So independent data, evidence of faster and more significant aging. But what is the role? And by subjecting tissue culture media from stem cell lines, Alex identified a number of proteins which were either specific for control cell lines, which were absolutely exclusive for progressive MS cell line condition media. And one of them, HNGB1, which is an alarmin, and one of the factors associated with aging, was again found significantly upregulated in MS cell lines, which went down after treatment with rapamycin, because of the non-action effect of rapamycin on aging, and which again Alex has been able to replicate independently once in Cambridge, using much less invasive and much more compelling human NSC technology. What is the role of HMGB1? We have no idea yet in full, but what is important is that the very same SOX2 expressing progenitor cells in the MS brain, but not in the control brain, indeed express much more HMGB1. And that in vitro, the inhibition of HMGB1 with neutralizing antibodies is able to rescue a functional deficit of terminal differentiation that Alex was able to observe in OPC, oligodendrocyte cell progenies arising from the parental stem cell lines. This is absolutely exciting, and there is very strong effort now in the lab to understand more and to understand also the basic mechanisms of regulation of these neural stem cell aging in the progressive MS brain by combination of high throughput metabolic, genetic, and epigenetic screens that we are organizing, we are putting together with partners spread between Cambridge and Europe, France, and Italy, with the ambition of understanding what is wrong in the biological clock of the MS brain and to identify also a solution for this. The second strategy, the second combination of strategy that I wanted to discuss with you was it's of course what has been a major program of investigation in the lab for the last 15 years, even more than that, which is the transplantation of exogenous stem cells as a way to rescue the loss of tissue integrity and the loss of function in the brain affected by chronic neuroinflammatory conditions. And I guess that this movie is gonna tell you in a very few fractions of time, pretty much 15 years of work, starting with the idea of delivering therapeutic exogenous stem cells via minimally invasive biological routes. We have been testing over the years the intravenous injection, the intrathecal injection. Cells have been also injected, sometimes also subcute. And each of these time we have wondered about the capacity of injected stem cells to recapitulate some leukocyte-specific pathways, to interact with epithelia, and to enter the brain and to promote a multitude of actions, which range from the promotion of endogenous but also exogenous remyelination, as you can see from the video, the ability to protect and preserve axonal function directly or indirectly, but also the remarkable capacity to modulate host immune responses. And that's why we proposed a pretty provocative rescue what is partially lost because of the occurrence of this chronic inflammatory injury to the brain and spinal cord. However, after over 15 years of work, we also identified that there were still some major challenges to stem cell therapies for complex tissue-specific conditions, and of course we have a special interest and focus in regenerative neurology and neuroimmunology. And these challenges applied to the starting material, because we all agree that we need cells which are scalable, which are stable, which are non-immunogenic or non-tumorigenic or terogenic. So they have to be safe and scalable. We also need to identify what's the best way to deliver them to patients, even though the preclinical work that we and others have extensively done goes in favor of multiple routes of cell injection, but we need to provide feasibility in humans. We are still pretty much in the dark, but I'll show you a very neat example that we have published recently. We are still pretty much in the dark about druggable mechanisms of actions with evidence of paracrine effect played in vivo by transplanted stem cells. And also we need to identify what is the ideal number of cells and the ideal stage at which to transplant them. And finally, we need biomarkers looking for distribution, functional effects, survival, interaction with the host. Based on this list of priority of challenges with Luca Peruzzotti in the lab, and in collaboration with Frank Edenhofer in Austria, we have developed a very straightforward and translational program of investigation, where by refining the most exciting concept of cellular reprogramming, regenerative neuroimmunology, we applied the direct reprogramming by which somatic cells are directly used into some stem cells to the development of an innovative therapy for the treatment of progressive MS. Luca has been absolutely fantastic in this journey. So we started simply by developing strain-specific directly induced neural stem cells, which were injected into mice with full-blown MOG-induced experimental autoimmune encephalomyelitis model of chronic progressive multiple sclerosis. And we observed a significant amelioration of clinical and neurological deficits, which, however, was absolutely super important of stem cells with the old generation controls. And there was, of course, no effect provided by the injection, in particular of the starting material, which you can see here in orange as fibroblasts. Integration survival was absolutely remarkable, with total of between 5 and 8% of surviving cells up to 110 days post-transplantation. The pictures that we took were absolutely brilliant. What was unexpected was to see such a massive diffusion and colonization of the host brain that you can see from the 3D neuro-elucidate-based reconstruction of the green cells after a single left lateral ventricle intrathecal injection. So cells in vivo were able to travel even for very long distances. Some of them were able to still to proliferate. There was no evidence of teratoma and masses, and they were able to colonize massively the brain and the spinal. At this stage, everything was pretty much published, because we had published before the very same evidence using in previous experiments fetal neural stem cells. However, looking at inflammation by an ex vivo multicolor flow cytometry of CNS infiltrating cells, we found no effect of the transplantation on antigen-specific adaptive immune responses, so no effect on T cells and B cells, but rather we found a significant change, not in the number, but in the molecular phagocytes. Looking at comparison between white and black or gray bilymphomas, you can see a significant reduction in the proportion of CX3CR1, but also CCR2, microglia and macrophages, expressing CD80 as a costimulatory molecule reminiscent of pro-inflammatory phenotype. So reduction of the pro-inflammatory phenotype and increase of the anti-inflammatory phenotype. So there was a significant switch in the phenotype of these myeloid infiltrates in the brain and spinal cord towards an M2-like anti-inflammatory alternatively activated pro-tissue-eating phenotype, which was absolutely unexpected. So at this stage, we had no idea why all this was happening in the absence of a major mechanism of action. So we started discussing with Christian Fretz in Cambridge, who gave us the suggestion of treating the experiments in a very agnostic way. So we designed this screening in vivo work in a way which is pretty much similar to what we do in the clinical practice when we have to deal with a patient with MS, looking at the blood and looking also at the CSF, because there is very strong evidence that the CSF holds the secret of a disease which affects the brain and the spinal cord, but without very much interaction with the periphery. So we did EAE, we induced EAE, we transplanted our mice with control and stem cell lines. And then we collected plasma and CSF early and late, 10 and 30 days post stem cell therapy. And we subjected matched samples of plasma and CSF to untargeted metabolomics by revealing a number of metabolites which were fluctuating and showing increase or decrease, paralleling their behavior in the CSF and plasma. There was only one small metabolite which showed a significant increase in the CSF only 30 days post disease induction, and which was instead significantly reduced following the transplantation of both fetal and directly induced neural stem cells. And that metabolite was succinate. You see how it does change in the CSF, but we know changes in plasma. So that means that succinate is fluctuating only in the CNS, in the CSF, and it is responding, its levels are responding to the transplantation. And in fact, also in vitro, when looking at the interaction between the stem cells and the very same myeloid cells which use succinate to switch from an M0-like quiescent-like to an M1-like pro-inflammatory phenotype, we found pretty much the very same effect, where co-cultures between stem cells and macrophages were able to induce a significant reduction in the extracellular acidification rate, while paralleling an increase in the oxygen consumption rate, which is reminiscent of reduction of glycolysis and increase of OXPHOS, respectively, compared to controls. But what is the role of succinate, and if succinate does or does not play a role in this? And the answer is potentially yes, because succinate is a metabolic signal in inflammation. Its cognate receptor is deorphanized. It's called SACNAR1 or GPR91. It is widely expressed on immune cells, including dendritic cells. Succinate, upon binding with GPR91 or SACNAR1, is able to signal intracellularly, inducing increase of IL-1 beta through IF-1 alpha. And the extracellular accumulation of succinate following reperfusion injury takes place through mitochondrial rows. And this GPR91 on cells is able to sense extracellular succinate to exacerbate tissue-specific inflammation. All this information was important because then we needed to fill a gap between the indirect correlative evidence of increase of succinate before and decrease after transplantation, and the role of succinate in the anti-inflammatory effect that we were observing following stem cell transplantation. And the missing information was that by chance, neural stem cells, human, mouse, fetal, directly reprogrammed, do express significant levels of GPR91, which is functional, because if we treat neural stem cells with succinate, it does signal, as we see here by calcium. So there is potential for GPR91 on neural stem cells to be sensing the excess of succinate that the CSF was showing as a consequence of the induction of EA. And in fact, the consequential next step was to develop SACNAR1 loss of function neural stem cells to see what they do. They are absolutely fine. They are expandable. They are multipotent in vitro after growth factor withdrawal. And of course, they fail to signal to succinate when treated with succinate. But they are also unable to scavenge succinate out of tissue culture media in an in vitro succinate uptake assay, as compared to wild-type fetal and directly induced SACNAR1 expressing controls. But in vivo, they fail. They still ameliorate EAE, but they fail to ameliorate EAE to the very same extent as wild-type SACNAR1 expressing neural stem cells do. And the very interesting thing is that by looking at the myeloid cell infiltrates, the lack of SACNAR1 on neural stem cell surface make them practically unable to induce the very same phenotypic switch from M1 to M2-like myeloid cells type transplants do. And also that they fail to scavenge and reduce the levels of succinate from the CSF, but of course having no effect on plasma. So this is what we have observed in mice, but it is not everything, because we also know that this interaction between the brain and the immune system, which can be mediated also by stem cell graft, can also be mediated by a significant recycling membranous components that each cell is able to release in the extracellular compartment in nature, which is something that we call generally extracellular membrane vesicles. And we came to this point after a few years of work and after having demonstrated that neural stem cells, exosomes and EVs exist. They are able to traffic functional cytokine signal. Also, they are immune competent, and they also can function as but the concept of regenerative neuroimmunology and the interaction between stem cells and myeloid cells. What led to engage into an additional round of very, very complicated experiments was that by looking by means of untargeted tandem mass tag based proteomics and the proteins expressed by EVs and parental stem cell lines, we found a significant enrichment to mitochondria proteins, or mitochondria-associated proteins in EVs compared to neural stem cells. So we wonder whether there was an additional thing to consider in this interaction, which was mitochondria. And in fact, by elaborating further these proteomic analysis, we found a significant enrichment in all the proteins belonging to the three major structural components of mitochondria, including the outer membrane, the inner membrane, and matrix in EVs compared to parental cell lines. So it was not just a random finding. It seems to be very consistent. And in fact, looking in more details, proteins coded in the mitochondrial nuclear genomes were all significantly enriched in the gray bars, which are the EVs compared to neural stem cells. So at this stage, the question was, is this a contamination of the exosomal EV preparation, or it is something that goes beyond the possibility of having it contaminated by cellular fragments? And we look for ND1, which is encoded in the mitochondrial DNA, and SDHD, which is instead encoded in the nuclear DNA. The absence of nuclear components, including histones, is a major feature in favor of the purity of your EV or exosomal preparation. And what we found was absence of nuclear genome, but instead we found in EVs and also in purified mitochondria a significant presence of ND1, which is suggestive of presence of mitochondria with intact matrix into EVs. So the next stage was to get crazy and to be absolutely paranoid. That's what Luca and Corey in the lab did for a few months by comparing custom-made, personalized, commercial kits, protocols for exosomal collection and EV preparations. And any protocol that we tested was producing a significant, strong evidence of all the 5 mitochondrial complex proteins in exosomal fractions, but also in EV fractions. And you see here 1, 2, 3, 4, 5 different protocols that we tested. So it was consistent and it was not protocol dependent. So mitochondrial proteins are present in exosomes and EVs. And in addition to mitochondrial proteins, we also find mitochondrial structures. And we call mitochondria-like to be cautious, but indeed, they are extracellular, they are intact, and they are membranous with a double membrane mitochondria. In fact, by applying a combination of cryo-EM with immunogold and other techniques, we provided an average size which falls right in the middle of what eventually is coded as EVs in total. We provided evidence of co-expression of the mitochondrial protein TOM20, and also we provided evidence of depletion or enrichment when comparing mito-depleted and controlled total EVs by flow cytometry. So there is indeed a significant, strong presence of about 30% of the particles of structurally intact mitochondria. And then at this stage, the question was, what about the function? What is the role of these mitochondria, which cells are either trafficking or releasing extracellularly, which are surrounded by double membranes, and what do they do on target cells? So we started from basically from characterizing the mitochondria, and we concluded that mitochondria have a mitochondrial potential, which is responsive to an uncoupler, CCCP, same way as absolutely pure fractionated mitochondria. And they also are able to breathe because they consume oxygen when substrates for the mitochondrial complexes are provided to the whole EV preparation. So they are structurally intact and they are functional intrinsically. But what about on target cells? So we started from a very extreme experiment that we designed together with Carlo Viscomi, at that time still working in MSc mitochondrial biology unit in Cambridge, now back in Padua, in Italy, where we challenged ROD0 cells with extracellular mitochondria. These ROD0 cells are very interesting because they are completely depleted in mitochondrial DNA, and they show an auxotrophic growth and overall survival, which is dependent from the extracellular uridine. So basically, they die spontaneously unless they are provided with uridine because they lack mitochondrial DNA. And we believed that we could provide exogenous mitochondrial DNA by means of EVs. And in fact, first of all, we checked for the incorporation 3 hours post, and we found a much larger incorporation or uptake of EV-associated mitochondria as compared to isolated mitochondria. 3 hours. Then 5 days, we counted the cells and we found, looking at 2 different dilutions, we found a significant increase in the number of surviving cells in the mitochondria-treated conditions compared to controls. And then we look at 16 days because we wanted the molecular confirmation of what was happening, whether it was a bystander effect or whether it was specific. And in fact, by sequencing the DNA of these ROD0 EV or Mito-EV treated cells, we found evidence of NTND3, which is a subunit of the respiratory chain complex 1 that was found absolutely integrated in the host genome. And this was the final proof for an efficient mitochondrial DNA integration in target cells with a complete correction of an intrinsic experimentally artifactual mitochondrial DNA function. But it was not enough because, of course, ROD0 cells do not exist in nature. So we wonder what was going to be the impact of these very cool but still difficult into more biology. We reminded ourselves that LPS activated mononuclear phagocytes become mitochondrially dysfunctional because of these shifts in their TCA cycle. And therefore, they might represent a nice host, target cell to challenge the Mito-EV hypothesis. And in fact, in this context, by treating LPS with LPS mononuclear phagocytes and then exposing them to Mito-DS-RED label EVs, we found, first of all, an efficient integration of these exogenous EVs into the host mitochondrial network by morphology, by STED microscopy, and by transmission electron microscopy. But also we found significant rescue of the glycolytic respiration, which is instead specific for pro-inflammatory LPS activated macrophages, with reduction of the oxygen consumption rate and induction of the extracellular acidification rate. So basically, what was provided by these Mito-DS-RED extracellular vesicles very much enriched into mitochondria was a rescue effect on the intrinsic LPS-induced dysfunction, respiratory dysfunction of activated macrophages. And then the final experiment was, of course, to go in vivo and to do the same in an animal disease model where this metabolic shift from M0 like resting into M1 like pro-inflammatory, highly glycolytic or OCRI macrophages is highly pathogenic. So we induced again EAE in mice, and then we injected ICV, a single preparation of either whole neural stem cells transduced with a lentiviral vector coding for FGFP and Mito-red, or EVs, different controls, total EVs, EVs depleted by mitochondria, EV depleted by CD63 immune-reactive subfraction of EVs. And what we found was not only that the white and the gray, which is the parental neural stem cell lines, and the total EVs have a remarkable rescue effect on EAE, which is pretty much super impossible, but that if you deplete your EV preparation either of mitochondria in blue or in CD63 immune reactive subfraction of EVs, you lose very much of the therapeutic effect. And in vivo, we found very diffuse, yet predominant in mononuclear phagocytes, the F480 immune-reactive cells on the far right, uptake of the Mito-red injected particles, which are the EVs that we have been studying so far in vitro. So overall, this is pretty much 20 years of work with evidence of multiple interaction and functional effects when looking at the crosstalk between the brain, the spinal cord, the cells, of endogenous reparting mechanisms, neurotropic support, metabolic reprogramming of target cells, inhibition of reactions to the injury, including astrogliosis and microgliosis. However, still, the annoying question is why to use stem cells if they are not intrinsically regenerative. So together with Robin Franklin and Charles Zhao, we decided to design another experiment to challenge specifically the question of whether stem cells of the brain are regenerative, intrinsically regenerative in vivo after transplantation. And the idea came from this isolated finding of two GFP exogenous transplanted in white, the transcription factor OLIG2, which is reminiscent of an early commitment towards an oligodendroglial in vivo in the corpus callosum of EA mice, very close to two areas which I have highlighted by the dashed lines where myelin has been damaged. As you can see here, in fact, there is a significant reduction of the intensity of the red fluorescence, which stands for myelin basic protein. So close to areas of demyelination, they seem to be taking a cell identity pathway which goes towards degeneration of new malforming cells. So with Robin, we designed a focal experiment. So we induced lysolecithin-mediated MS-like lesions or induced new stem cells. And we found that 21 days post lesions, cells survive. They predominantly remain around the lesion where lysolecithin was able to induce a chemical demyelination. And interestingly, they differentiate, they progress towards an OLIG2 CC1 positive oligodendrocyte program of differentiation. And interestingly, they provide more than twice as much new myelin to the host, with a significant proportion of these newly generated myelin-forming cells belonging to the endogenous GFP negative compartment, but at least 1/4 of them are coming from the graft. And with also evidence of differentiation into PLP expressing mature oligodendrocytes. However, within this context, we cannot exclude that part of the data that we have collected might be biased by the fact that there would be a significant level of competition for naked axons between the endogenous pool of progenitors recruited in situ and the exogenous transplanted stem cells differentiating in vivo into myelin-forming cells. So we developed another approach by which the very same experimental model was induced into OLIG1 knockout immune-competent mice, which are described to have a significantly delayed endogenous remyelination, therefore reducing the risk of the cellular graft to compete with endogenous OPCs to make new myelin around the axons. And also in this case, we found that when comparing NSC transplanted versus PBS injected oligonucleotide mice, we found evidence of exogenous exogenous remyelination, therefore implying differentiation of exogenous cells towards mature remyelinating oligodendrocytes with significantly thinner myelin. And the final strategy, which I'm gonna very, very briefly touch on in one slide, is that technology is available to put all these principles together and to apply in vivo the focal injection of either integrating or non-integrating or soluble small molecules, which will be targeting post-mitotic cells, inducing efficient, still not very efficient, but at least as proof of concept, this evidence is available, efficient reprogramming of brain-specific somatic cells into a desired cell of interest with multiple potential applications to different conditions, including progressive MS. This is the very next future, and I hope there would be additional chances to discuss of the in vivo reprogramming data in the next future. So just to go quickly to the end of my talk, I think I have provided you provocative but also compelling evidence of which are the major challenges for developing new generation molecular medicines in regenerative neuroimmunology, with a specific case study that is progressive MS, by showing that mitochondrial metabolism in immune cells might be a major target, that route of delivery of therapeutic vehicles needs to address multiple lesions. So the intracerebroventricular / CSF route might be a good way to go. That the chronicity of the disease is important to consider when prospecting an ideal window of opportunity for patients, and therefore ideally targeting a stage of the disease which is transiting or transforming from a relapsing, remitting early stage into a secondary progression would be a good option. That regeneration in vivo requires semantically terminal differentiation and functional integration, but this might not be absolutely necessarily to rescue a lost function. That the immune activation of the host, which can be sometimes paradoxical, might not be necessary for non-immunogenic autologous or autoidentical stem cell grafts. That we have to consider that the immune system of the host might be inhospitable to molecular medicines of any type. That there might be some intrinsic deficiency in the stem cells from the patients, and that I guess what's intrinsic flexibility of stem cells to progress towards functional states where anti-inflammatory or neuroprotective of intrinsic direct regenerative capabilities become predominant. Therefore, having stem cells working as a natural occurring full combination therapies. And this is the most important slide of my talk, where I would like to thank the members of my lab, present and past, who have provided a significant contribution to the data that I've shared with you today. So I have to thank Luca Peruzzotti Ametti, who has been a major driver in the lab, Alex Nicaise, Corey Willis. They are very much into the aging program. Bristena Ionescu is an CDTP student looking at aging, working specifically the role of SACNAR1 in myeloid progenitor cells, Regan Hennel, and also our lab alumni, and our collaborators in Cambridge and abroad, Robin Franklin in Cambridge, Carlo Viscomi, Massimo Zeriani, Mike Murphy, Alain Brisson, Valentina Fossati, and Frank Edenhofer. And I thank you very much for your attention. I'll be very happy to take up as many questions as you like.
Thank you, Stefano, for that great talk. That's it for this term. We hope you've enjoyed it. Join us back again in October 2021, when we will bring you more of the cutting edge and interdisciplinary research that is taking place across Cambridge Neuroscience. In the meantime, for all things neuro related in Cambridge, follow us on Twitter at CamNeuro. See you next time.